Greetings to everyone. Hope you all are hale and hearty. And we are back with another module in continuation with the previous module on solutions to sample paper number 1. And in this module, we're going to be addressing the question number 16 to question number 20. And I am Ujaswata Pridarshini Gaur, an undergraduate from MBM Engineering College, Jodhpur. So without any further ado, let's get started. So the first question says, we're going to divide, okay, now I want you to understand the approach to this question. We're going to divide each question into two parts. So the first part is define the term with an example of symbiosis, parasitism, and autotrophs. And then the second part, we're going to be identifying the plant group with stems and leaves, but with hidden reproductive organs, and stems and leaves, but with no vascular systems for conduction. So the first part says, in which we are going to define what is symbiosis, what is parasitism, and what is autotroph. So I want you to have a closer look at this picture right here. Now, this is going to give you a clear understanding of what symbiosis, communalism, and parasitism is. Firstly, symbiosis is a close living interrelationship between two organisms of different species where one or both of them are benefited from each other's coexistence. So, right, if you can understand this mutualism, this is nothing but symbiosis as well. In this, the flowers, they get pollinated, and these bees are going to sit on these flowers and they collect nectar, and in turn also these flowers get propagated from one place to another place, so their generation continues, their growing continues. So in this way, both of these genes, both of these species are benefited from each other, so it's a mutual relationship, it's a symbiotic relationship. The other example can be fungus, which is growing on the roots of host plants. The fungus will help the plants in better nutrient absorption through the roots and also protects the plants, right? In parasitism, it's only one way, like in the parasitic plants, they get all the nutrients from their host plants, but they do not benefit the host plant in any way, and sometimes may even damage the host plants. The examples, these are cascadas, and if you want to understand it in a better way, for example, you can understand that from a cat, it gets a skin irritation from the bugs or the ticks that are fed on the cat's blood. So this is a one-way process where the tick is benefited from the cat's blood, but then the cat is not benefited from the tick in any way. In fact, it gets an irritation, so it's very harmful for the cat. So this is a parasitic relationship. Now, what are autotrophs? Autotrophs are nothing but plants that produce their own food with carbon dioxide and water with the energy from the sunlight. So these are all green plants and green algae and bacteria. Any plant which is able to manufacture its own food is nothing but an autotroph. Now, the second part of the question says, in which we have to identify the plant group with, first is stem and leaves, but with hidden reproductive organs. Then the answer to these questions are nothing but pteridophytes, and the stems and leaves, but with no vascular systems for conductions, are bryophytes. So this is an example of a pteridophyte right in front of your screen. And bryophytes, these are bryophytes in which they are non-vascular, they depend on easy access to water, and they have restricted height without fortified tissue for support. Okay. Now, let's move on to the second question, which says, again, divided into two parts. In the first part, we'll calculate the acceleration required to push a box weighing 3.5 kgs with 2 newton of force. And then a B part to the part A says a carrot weighing 45 kg was pushed on the floor to about 15 meters with a force of 20 newtons. Calculate the work done. In the or part, we have there is a change in the velocity observed from 10 meter per second to 15 meter per second when a force of 300 newton is applied on a body. Calculate the mass of the body. And in the B part, we have what would be the force required to accelerate the speed of a vehicle weighing 1500 kg moving at a speed of 10 meter per second square. So in the first question, wherein first we have to calculate the acceleration required to push a box weighing 3.5 kgs with 2 newton of force, I want you to understand in this we have been given that we have a 2 newton of force that is going to be our F net, that is going to be our force. And then we have the weight, that is the mass, is also given to us to be 3.5 kg. So when we apply this formula, that force is equal to mass into acceleration, which is nothing but the Newton's second law. We're going to substitute the value of F to be equal to 2 newtons, A to be equal to 3.5 kgs. A is what we need to find, and the force is 2 newtons, the mass is 3.5 kgs, and then we have to find the value of A. So that can be achieved simply by dividing the F with M. that is 2 Newton over 3.5 kgs, and then you get the value of A to be 0.57 meter per second squared. In the next part, we have a crate weighing 45 kgs pushed on the floor to about 15 meters with a force of 20 newtons. Calculate. So in this picture, you can see, let us assume that this is the crate, which is 45 kgs, and then this has been pushed on the floor to about 15 meters with a force of 20 newtons. So this person is applying a force of 20 newtons here. So first we need to understand that work can also be written as force into displacement, and the displacement in this case is also given to us to be 50. So the work done can be easily found out by 20, that is the force, into 15 meters, that is the displacement, and that easily gives us the value of W to be equal to 300 joules. The only trick was this formula, W is equal to F into S, and keep in mind the units that you're using while calculating. In the part B, we have to find the change in velocity observed from 10 meter per second to 15 meter per second when a force of 300 Newton is applied on a body. Calculate the mass of the body. So when it says that it has been a change in velocity is observed from 10 meter per second to 15 meter per second, that means this is change in velocity is nothing but acceleration. If you go by the definition of acceleration, you have it to be the rate of change of velocity, and here it's directly given us to be from 10 meter per second to 15 meter per second. The change in velocity is going to be 5 meter per second, and this is going to be the change in velocity and rate of change of velocity. And then since we have the rate of change of velocity, that is nothing but acceleration, and then the force is also given us to be 300 newtons, we can easily calculate the mass of the object by applying the Newton's second law of motion, which is force is equal to mass into acceleration. And then henceforth we find the value of m, which is equal to 300 divided by 5, that is 60 kgs, and the value of mass is equal to 60 kgs. In the B part says, what would be the force required to accelerate the speed of a vehicle weighing 1500 kgs and moving at a speed of 10 meter per second squared? Okay, so we need to find the force in this case, which has an acceleration that has given to us to be 10, and then the mass is given to us to be 1500 kgs. This is a very simple example. Just to apply the second law of motion, the force is equal to mass into acceleration. The mass is given to you, the acceleration is given to you, so you can easily find the value of F, force, which is equal to this mass 1500 into 10 meter per second squared. That is 15,000 newtons. Keep in mind the values, the units that you're using, and change the units accordingly, whether you're using the SI system or the CGS system, the British system of units. And this is just a graphical representation of the relationship between force, mass, and acceleration. The force and mass are directly proportional to each other, and the slope of this curve between the force and mass gives you the value of acceleration. This is a direct relationship. That's how you get F is equal to m into A. So F is directly proportional to m, and when you remove the directly proportional constant, you get a value of constant that is equal to your acceleration. Now the next question says, explain the saturation and solubility of a solution. And the B part says, how do isotopes differ from isobars? Mention two medicinal uses of isotopes and represent the atomic number and the mass number of isotopes of chlorine and carbon. So in the first part, we're going to first try to have a closer look at what is solubility and saturation. So solubility of a substance or the solute is the ability to dissolve in a solvent. At a given temperature, if the solute completely dissolves in a solvent, its maximum capability, then you have the solution is said to be saturated solution. If the amount of solute is less than that of the saturation level, then it is said to be an unsaturated solution, right? Now from this, the concentration of solutions can also be determined, which is the amount of solute present in a given amount of solution. And this is represented by this formula right here. And in terms of volume percentage, it can also be represented. Solubility is just not to be confused with the ability to dissolve a substance. Now this is very important. It is not to be confused with the ability to dissolve a substance, because the solution might also occur because of a chemical reaction. The solubility of a substance is an entirely different property from the rate of solution, which is how fast it dissolves. The smaller the particle, the faster it dissolves. And there are also many factors to add to this generalization. And right in front of you in this picture, you will observe there are four solutions. One is a saturated solution, one is a unsaturated, then you have a saturated solution, then you have a supersaturated solution, and then finally you have a precipitate formation. So in the unsaturated solution, here is from where you can understand the concept of saturation is that when you were adding this solute particles of this orange color into your water or any other solvent, it was dissolving, kept on dissolving until it reached the state of saturated solutions in which it was completely dissolved. Now, when you added a little more of this solute to this water or this liquid, it had attained a condition of supersaturatedness. So, what is supersaturatedness? If we add just a little bit of these solute particles more, they'll start precipitating out. So after this supersaturated state, you have a precipitate formation in which even if you add just a little bit of anything, it will have a precipitate formation right here. You can see this is the precipitate has already started forming, but till now we just need to take into consideration what is saturated solution. So yes, when the complete amount of solute has been dissolved into the liquid, which in the amount that it can actually hold, then that is what forms a saturated solution. Now the B part asks us to first, how do isotopes differ from isobars? Now isotopes are the atoms of the same element with the same atomic number, but with different mass number. That is the same atomic number but the different mass number. And here you have an example of hydrogen. It has three isotopes. They have protium, deuterium, and tritium, and the atomic mass of all the three is the same one, but then the mass numbers are all different. That is 1, 2, and 3 accordingly. Because then how does the mass number, because see, mass number is nothing but the number of protons plus neutrons, right? We have discussed this in the previous modules also. Mass number is nothing but protons per neutrons, and atomic number is the number of protons. So what is differing in each case of an isotope is the number of neutrons. See, when you have the number of neutrons are different, you get different isotopes of the same element, right? So for example, you can easily have protium, then you have deuterium, and you have tritium. In case of isobars, you can see these are isobars with a different atomic number but the same mass number. That is, your P plus N, your P plus N value is going to remain the same, but your P is going to differ for each element, right? And here are the examples of 40 sulfur, 40 chlorine, and 40 potassium. They all have the same mass number of 40, but they have the different atomic numbers, right? So this is the first part. The next part asks us to mention two medicinal uses of isotopes. Then these are the two medicinal uses. One is the isotope of cobalt, which is used in the cancer treatment, and the other one is the isotope of iodine, which is used in the goiter treatment. Now have a look at the screen. This is iodine 131. This is the isotope of iodine which is actually used in the goiter treatment, isotope 131 of iodine. And this is cobalt. This is the generalized cobalt, in which you can observe that it has an atomic number of 27. It has a atomic weight of 58.933 grams. It has a crystalline structure, and then it has an electronic configuration of argon with 3d7 and 4s2. It is a transition metal, and then it's just a solid state as a physical state at 20 degrees Celsius, and it is a hexagonal structure, right? And let's move on to the third part of the question, which says represent the atomic number and mass number of the isotopes of chlorine and carbon. So these are only two of them, and we're going to discuss only, they can be many, but here we're going to discover, we're going to read about only two of them. One is going to be the chlorine, which has 35 Cl17 and 37 Cl17. This is 35 is going to represent your mass number, chlorine, and then this is 17 is your atomic number. Now since these are isotopes, the atomic number is going to remain the same. That is 17 here, 17 here, but the mass number is different. That's why it has a 35 mass number here and a 37 mass number here, and this is why these are two different isotopes of chlorine. Similarly, in case of carbon, you can see the atomic number is 6 in both the cases, but then the mass number is 12 in one case and 14 in the other case. So these are isotopes with the same atomic number but different mass numbers. Now let's move on to the next question. It says, again, two parts. The part A says, what should be the distance from a cliff if a person needs to hear the echo of his voice after 5 seconds, provided the speed of the sound is 345 meter per second. And then B part says, what would be the velocity of a wave with frequency of 20 hertz and wavelength of 20 meters? Mention if the frequency of the sound is in audible range or not. Then we just need to understand here that The speed of the sound. What is the speed of the sound? It's given us to be 345 meter per second, and the time taken to hear the echo is also given us to be 5 seconds. So the distance traveled by the sound is going to be the velocity into the time. That is 345 into 5 seconds, it gives us 1725 meters. Now, in 5 seconds, the sound of the voice travels twice from the person to the cliff and then back to him. Hence, the distance from the cliff to the person is nothing 1725 divided by 2, that is 862.5 meters. And in the next part, where we have to find the velocity of the wave with frequency of 20 hertz and the wavelength of 20 meters, and mention the frequencies in audible range or not, then yes, the frequency is given us to be 210 hertz. The wavelength is given us to be 20 meters. Now, velocity is going to be wavelength into frequency. This is a standard formula. So when you put in the values of wavelength and the frequency in this formula, you get a value of the velocity to be 4,200 meters per second square, per second, sorry. And since we know that the frequency of the audible range is from 20 hertz, 20,000 hertz, this is detectable and audible to humans. And that's why the sound, the frequency that is given to us, that is 210 hertz, lies in this range, and therefore it comes to be in the audible range. This is audible. This is definitely perceivable by humans. Let's move on to the last question, which is again divided into two parts. Mention the function of the following cell organelles. The first is the Golgi apparatus, then you have mitochondria, and then in this figure we have to identify the cell organelles named A and B. Then we have to differentiate between the two of them and then mention each function of them. And in the second part, we have to draw the diagram of a prokaryotic cell, explain how prokaryotes are different from eukaryotes with respect to the cell division, and then draw a neat and label diagram of a phloem cell, and then name that phloem cell that is tubular in structures. Okay, so let's move on to the first part, in which the first part's first question, which says mention the functions of the following cell organelles. The first is the Golgi apparatus. So Golgi apparatus is an organelle which is present in most eukaryotic cells. It is made up of a membrane-bound sacs that is also called a Golgi body or a Golgi complex or dictyosomes. They are made up of sacs called cisterns, and they work closely with the rough ERs. And the functions of Golgi apparatus is what? They help in the storage of protein. They modify the sort and packs of proteins, and they create lysosomes. And this is a structure of a Golgi apparatus. You might observe they have cisterns, they have myoforming vesicles, and they have the secretory vesicles, they have lumens, and they have incoming transport vesicles here. And this is how it looks inside a body of the cell. The second part is about the mitochondria, in which a mitochondria is nothing but the rod-shaped organelle that can be considered as a power generator of the cell, converting oxygen and nutrients into adenosine triphosphate molecules. These are energy currencies of the cell that provides power to the cell's metabolic activities. And this process is also called aerobic respiration. Since see, you can understand this better by understanding that why is it an aerobic respiration, because it is involving oxygen right here. If there is no oxygen here, it becomes an anaerobic respiration. So it is an aerobic respiration because it has oxygen, and then it is breaking down these oxygen and nutrients to what is called as adenosine triphosphate or ATP molecules, which are the energy currency of the cells, which are utilized for cells metabolic activity. So this is the powerhouse. This is rod shaped, and then if you look at the structure, you have some cristae, you have matrix, and then you have an inner membrane and an outer membrane. And the functions of mitochondria will include definitely production of ATP, that is the energy currency of the cell that we discussed, and a cellular metabolism as well. Now the B part, the second subdivision of the A part, is wherein we have to identify the cell organelle of this diagram, this A and B. What is this A and B? Then this A is the the rough endoplasmic reticulum, and B is the smooth endoplasmic reticulum. The difference between RER and SER, that is the rough endoplasmic reticulum and the smooth endoplasmic reticulum of a cell, is as below. Now, the smooth endoplasmic reticulums have no ribosomes, whereas the rough ones have ribosomes. That tubules are present in smooth ERs, whereas cisterns are present in the rough ERs. The smooth ERs are located near the cell membrane, and the rough ERs are located in the cytoplasm. And finally, the smooth ERs are formed from shedding of the rough ERs, whereas the rough ERs are formed by the nuclear membrane. And now this is a final structure. This is the complete figure of that. This is the nucleus, this is your nucleus envelope, and this surrounding this is your rough endoplasmic reticulum. This has cisterns in it, as I told you. This is a cistern structure. And then you have tubules here, you have tube-like structures here. These are tubules, and that is why these are smooth endoplasmic reticulum right after your rough endoplasmic reticulums. And the third part says, explain how prokaryotes are different from eukaryotes with respect to cell division. Oh, no. Mention one function of each of them. So, endoplasmic reticulum is an organelle found in both eukaryotic animals and plant cells. It often appears as the two interconnected subcompartments, namely rough ER and smooth ER. Both are consisting of membranes, and they are interconnected flattened tubes as well. Now, when you talk about the rough ERs, they're studded with millions of membranes bound ribosomes, and they are involved in the production and folding control of quality control and dispatch of some proteins. They can synthesize and store lipids, whereas smooth ERs can synthesize and store proteins. And this is the major difference. Smooth ERs are storing and synthesizing proteins, whereas rough ERs are synthesizing and storing lipids. And these both proteins and lipids are the biogenesis of cell membranes, and they are also called enzymes and hormones. The part B of the question asks us to first draw a neat and label diagram of a prokaryotic cell. So this is a prokaryotic cell. You have a bacteria flagella here, which is used for the movement of this prokaryotic cell. Then you have cytoplasm, in which all these bodies are embedded. You have ribosomes, you have a cell wall, you have a mesosome right here, you have a DNA, and you have a plasma membrane. And then you have this DNA nucleoid. This has a capsule shape. And yes, now the second part says, what is the basic difference, or explain how prokaryotes are different from the eukaryotes with respect to cell division. Then the biggest difference between the two of them when it comes to cell division is that prokaryotes are undergoing mitosis, whereas eukaryotes are undergoing meiosis. Now, cell division is simpler in prokaryotes and then in eukaryotes. So the basic difference is right here of mitosis and meiosis. And in the B part, we have to draw a neat and label diagram of a phloem cell and then name the phloem cell that is tubular in structure. So this is the diagram here of a phloem cell. It has a sieve plate right here, then it has a companion cell, then it has a sieve tube here. This is the sieve tube element, and then it has cytoplasm filled here. So this is your phloem cell. Now this is this is the top view of a sieve plate. This is how it looks like, and then you have all these vessels coming in from these holes, and they're hearing the transportation is going to take place of water and nutrients. And then the phloem cell that is tubular in structure is called as sieve tubes. And sieve tubes are these are tubular in structure. They're cylinders, and they're composed of elongated thin walled cells placed end to end. You can see this is one cell that is placed end to end to another cell, and the main function is to transport sugars and nutrients up and down, and this is what it actually looks like, right? So with this, we come to the end of the module. Thank you very much to be a patient audience. Hope you were able to understand the concepts well. And as I tell you every time, go by the fundamentals and you never go wrong. So keep learning, keep growing. Thank you very much. And now we'll meet in the next module.